Adult humans are made up of approximately 100 trillion cells. However, every second, millions of these cells die. Fortunately, every second, cellular division creates millions of new cells to replace the millions that die. In younger developing humans, cellular division not only replaces spent and dying cells, but also provides for growth and development by producing far more cells than are destroyed by life processes. Occasionally, the process of cellular division goes on uncontrollably, resulting in the illness we call cancer. In order to conquer cancer, researchers must understand the mechanisms that control cellular division and how these mechanisms are overridden by cancer cells. Information gained through research is constantly leading to more effective treatments for one of the most dreaded illnesses of our time. A more complete understanding of how cells reproduce is leading to other advancements in medical science as well. For example, recent discoveries suggest that in the future, doctors will be able to grow new nerve cells to replace those damaged by disease or injury, a prospect that looked very unlikely 20 or 30 years ago. Likewise, it looks as if eventually medical science will reach a point where hearts, livers, kidneys, and other organs can be grown specifically for transplantation. However, such a future will only become reality if researchers learn more about how cells grow, divide, and interact in order to form complex organs such as hearts and lungs and complete organisms. When cells divide and reproduce, they must pass life's essential ingredients on to their daughter cells. The genetic information contained in molecules of DNA and sufficient proteins, carbohydrates, lipids, and other molecules to enable the daughter cells to begin to carry out the functions of life. There are two basic types of cells: prokaryotic cells, whose relatively small amount of DNA is found in the cytoplasm along with the other ingredients of life, and eukaryotic cells, whose larger amounts of DNA are separated from the rest of the cytoplasm by a nuclear envelope. These differences in the amount and the accessibility of their DNA, along with others, leads to significant differences in cellular division and reproduction between prokaryotic and eukaryotic cells. Eubacteria and archaebacteria are the only living organisms to have prokaryotic cells. All other organisms, including protists, fungi, plants, and animals, have eukaryotic cells. Let's now look at how both prokaryotic cells and eukaryotic cells divide and reproduce, starting with prokaryotic cells. The complete sequence of events in a cell, from one cell division to the next, makes up what is referred to as the cell cycle. The cell cycle of prokaryotic cells can be very rapid under favorable conditions. For example, the intestinal bacterium E. coli can complete its cell cycle in 30 minutes or less. The majority of the cell cycle is spent absorbing nutrients, growing, and replicating DNA. The genetic material, or DNA, of most prokaryotic cells is contained in a single strand of DNA called a chromosome. The chromosome of a prokaryote is usually coiled in a circular shape and attached to a point on the cell membrane. The process of cellular reproduction begins with the chromosome duplicating itself. After chromosome duplication is complete, the new chromosome attaches itself to a point on the cell membrane close to the original chromosome. At this point, the cell wall and membrane start growing between the attachment points, pushing the chromosomes apart and elongating the cell. After the chromosomes are well separated from each other and the bacteria has reached about twice its initial length, the cell wall and membrane pinch inward, dividing the two ends into two new daughter bacterium, each having its own chromosome and about half of the cytoplasm. In prokaryotes, the process of cellular reproduction is called binary fission. Let's now look at the more complex process of eukaryotic cellular reproduction. The cell cycle of eukaryotic cells, which is typically much longer than that of prokaryotic cells, is divided into two major phases, interphase and mitosis, or the M phase. Interphase is the stage in the cell cycle in which a cell acquires energy and nutrients from the environment, grows, and duplicates its chromosomes. During mitosis, the chromosomes duplicated during interphase are separated to form two new nuclei. The later stages of mitosis are usually accompanied by a division of the cytoplasm into two daughter cells in a process called cytokinesis. Let's look at each of these phases in turn, starting with interphase. Cells often spend 90% of, or even their entire lives, in the interphase portion of the cell cycle. Throughout interphase, cells acquire energy and nutrients from the environment, grow, and carry out their specialized functions. However, a number of significant events occur during interphase that has caused biologists to divide interphase into three subphases: G1, which stands for first gap, S, which stands for synthesis stage, and G2, which stands for second gap. G1 is the phase after the most recent cell division and before the duplication of chromosomes starts. A large percentage of a cell's growth usually occurs during the G1 phase. However, some cells, such as the first cells to divide in animal embryos, do so very rapidly, with little or no growth between divisions and virtually no G1 phase. Later in the G1 phase, at a time referred to as the restriction point, cells undergo an internal evaluation of their ability to complete the cell cycle and produce two viable daughter cells. If the evaluation is negative, the cell does not go on to the mitosis phase, but rather exits the cell cycle to enter a non-dividing state called the GO phase. If the evaluation is positive, the cell becomes committed to DNA replication and mitosis. The restriction point can also be thought of as a point of no return for cell division. If a cell is going on to mitosis, it begins to produce proteins called histones that are critical to organizing and condensing chromosomes during mitosis. As the production of histones peaks, the S phase of interphase begins. During the S phase, duplicate copies of each chromosome are synthesized. This process can take quite a while, as many eukaryotes have a large number of very long chromosomes. For example, human cells have to duplicate 46 chromosomes, each 1,000 to 5,000 times the diameter of the nucleus in length when stretched out. After chromosomes are duplicated in the S phase, G2, which is usually the shortest phase of interphase, begins. During G2, cells further ready for mitosis by synthesizing various proteins and other molecules that will be required for both mitosis and cytokinesis. After G2, interphase is over and mitosis, or the M phase, begins. Mitosis is divided into four phases that tend to occur continuously rather than as discrete events: prophase, metaphase, anaphase, and telophase. As prophase begins, the chromosomes duplicated during the S phase of interphase, now consisting of two sister chromatids attached to one another at a junction called the centromere, lie loose and fully extended in the nuclei. Chromosomes are loose and fully extended during interphase so that they can be duplicated, and so their genes can be read by the rest of the cell in order to carry out cellular processes. However, in their extended state, chromosomes are very long and difficult to separate. Thus, during prophase, the first event that must occur is for the chromosomes to be condensed down to a manageable length. It was discovered by scientists in the early 1970s that DNA in the nucleus is wound around proteins called histones. A short section of DNA, wound about a group of 8 histone molecules, forms a nucleosome. Nucleosomes interact with one another to shorten and thicken chromosomes by coiling about one another and then coiling again to create supercoils. Condensed into supercoils, once invisible chromosomes become visible under a light microscope. As the chromosomes in the nucleus become visible, another structure in the nucleus, the nucleolus, begins to disappear. The nucleolus is involved in the synthesis of ribosomes, which, as you will recall, are small organelles that manufacture protein molecules. The dark area in the nucleus formed by the nucleolus is the result of a very heavy concentration of DNA, RNA, and proteins interacting to synthesize the components that make up ribosomes. As chromosomes condense and separate, ribosome synthesis stops and the nucleolus disappears. Later in prophase, a structure called a spindle begins to form in the cytoplasm. The spindle is a football-shaped array of microtubules that eventually separate sister chromatids. The spindle arises from organelles called centrosomes, or microtubule organizing centers, as they are sometimes called. During interphase, the pair of centrosomes located near the nucleus have an array of microtubules extending out from them. As prophase proceeds, a new larger array of microtubules that will make up the spindle begins to form between them. As the spindle continues to grow, the two centrosomes move further apart and to opposite ends of the cell. Eventually, the network of microtubules that radiates from the centrosome at each end of the cell grows to extend beyond the equator of the cell and into the nucleus. After the spindle is completely formed, the nuclear envelope rapidly disintegrates. The double membrane of the envelope breaks up into vesicles that resemble pieces of endoplasmic reticulum. The breakdown of the nuclear envelope allows microtubules from the spindle to invade the nuclear region and attach to chromosomes. The microtubules that attach to chromosomes in the nucleus are called kinetochore microtubules, as they bind to structures located near the centromeres of each chromosome called kinetochores. Other microtubules, called non-kinetochore or polar microtubules, do not attach to chromosomes at all, but rather retain free ends that overlap along the cell's equator. During metaphase, the kinetochore microtubules, running to the different sister chromatids from opposite ends of the cell, engage in what looks like a tug-of-war, with each group of microtubules pulling the sister chromatids to which they are attached toward their end of the cell. Eventually, after what looks like a jerky back-and-forth struggle, the kinetochore microtubules become equal lengths, and each pair of sister chromatids ends up lying midway between the two spindle poles and aligned along an imaginary plane running through the cell's equator, called the metaphase plate. Metaphase ends when all chromosomes are lined up along the cell's metaphase plate. Anaphase begins when the centromeres between all the sister chromatids divide and new independent daughter chromosomes are formed. The kinetochores of the new independent chromosomes now start to walk down their respective kinetochore microtubules toward the spindle pole, pulling the rest of the chromosomes along with them. The kinetochores and their attached chromosomes walk down the kinetochore microtubules as the result of the action of motor molecules made up of the protein dynein. As these motor molecules walk down the microtubules, the sections of microtubule just passed break off. At the same time that chromosomes are walking down the kinetochore microtubules, motor molecules attached to polar microtubules walk down the adjacent polar microtubules, causing the poles of the spindle to grow further apart. Anaphase ends once all the chromosomes reach the end of the kinetochore microtubules to which they are attached, and the polar microtubules have stretched the poles of the spindle as far apart as they can. Identical and complete collections of chromosomes now lie at opposite ends of the spindle, as the next phase of mitosis, telophase, begins. As the final phase of mitosis, telophase, begins, the spindle disintegrates. The vesicles that formed when the old nuclear envelope broke up during prophase coalesce around the two new groups of chromosomes and form a nuclear envelope around each. Once enclosed in a nuclear envelope, the chromosomes relax into their extended state, and the nucleolus reappears as the new nuclei begin to synthesize ribosomes and other materials. In most cells, cytokinesis occurs concurrently with telophase, enclosing each daughter nuclei in its own separate cell. The process of cytokinesis varies substantially between animal and plant cells. In animal cells, microfilaments made up of actin and myosin, proteins that are involved in muscle contraction and other forms of cell movement, form rings around the equator of the cell. These rings are attached to the cellular membrane. During cytokinesis, the rings contract and pull in the equator of the cell, much like the pulling of drawstrings, creating what is called a cleavage furrow. As the contractile rings become smaller and smaller, the cleavage furrow becomes deeper and deeper, until the parent cell is pinched in two, producing two completely independent cells. Plants have stiff cell walls that can't easily be pinched apart. Thus, the process of cytokinesis in plant cells is quite different from that in animal cells. Rather than microfilaments pinching off the cellular membrane at the equator, the Golgi bodies of both daughter cells bud off carbohydrate-filled vesicles that line up along the equator of the parent cell. These vesicles fuse together to form a structure called the cell plate that divides the parent cell into new daughter cells. The membrane material that formed the vesicles fuses with the existing cell membrane on either side of the cell plate to form a new continuous cell membrane in each daughter cell. The carbohydrates contained in the vessels form the middle lamella of the new cell wall between the two plasma membranes. Each of the two daughter cells then secrete cellulose between their new cell membrane and the middle lamella to form the primary cell wall. Many plants later secrete additional cellulose molecules and other polysaccharides beneath the primary wall, forming a thick secondary wall. The events of interphase and mitosis, like so many other processes in our bodies and cells, are orchestrated by regulatory proteins. Biologists are just now beginning to understand how these proteins interact to synchronize the events of the cell cycle. But they've known for quite a while that different types of cells often proceed through the cell cycle at very different rates. In our own bodies, the cells that form our skin and line our intestines are continually dividing in order to replace worn-out cells. On the other hand, the nerve and muscle cells of adult humans, whose functioning depends critically on established connections and interactions with other cells, usually exit the cell cycle entirely by entering the GO phase. For cells that remain in the cell cycle, the decision to divide or not divide is controlled by both internal and external factors. The major internal factor controlling whether a cell in the G1 phase passes the restriction point and enters the S phase and duplicates its DNA appears to be the cell's size. Scientists hypothesize that as a cell grows larger and larger, the original complement of DNA in the nucleus can't meet the demands of continued growth, and a regulatory mechanism controlled by numerous proteins permits the cell to pass restriction point and enter into the S phase of interphase. One external factor controlling whether a cell goes beyond the restriction point is the presence of special regulatory substances called growth factors. For example, before fibroblasts, which are critical to healing wounds in the body, can pass the restriction point, molecules of platelet-derived growth factor, or PDGF for short, must attach to special PDGF receptors on their cellular membranes. Specialized blood cells, called platelets, which contain PDGF, concentrate at the site of wounds and release PDGF triggering fibroblasts adjacent to the wound into cell division and repair of damaged tissues. The fibroblasts eventually stop dividing as a result of another external